Hello, in this video let us look at all the previous year questions that came in the anatomy final theory exam, okay, under histology. So what are we looking at? Histology. Histology is what? Study of tissue, right? So let us look at it now. So just the questions we are looking at. So here they are saying microscopic structure and of the transverse section of bone. What are they asking about? Bone. So what are they asking here? The transverse section. So you'll have to draw neat label diagram. So microscopy structure, that becomes histology. So just think, okay, they are asking you about the transverse section of bone, correct? So now let us assume that it is a long bone like this, okay? And here you're taking transverse section. So here when you take transverse section, what will you get? Ideally, what you will get is something like this, like a circle, right? So in this inside you'll have bone marrow, osteum and all that, right? Let's look at this. So this is a macroscopic structure, but okay, this is what? This is macroscopic structure. So outside what do you have? You have the periosteum. Then you have some Sharpie fibers. So here you have the compact bone. Then you have the endosteum. Then you have the medullary cavity here, which is filled with the red bone marrow in children and the yellow in adults. Okay. Did you get it? So this is not what you have to draw. What is this? This is macroscopic structure. But what have they asked you? The microscopic structure, correct? So then what will you have to draw? This is how the transverse section of bone will look under the microscope. Okay. So if you have to draw it, this is how you would draw it. So here you have the osteon. So this is one osteon. This is another osteon. This is another osteon. So this is bone cross section. And inside this osteon, in the center, you have this pipe going through, that is the Haversian canal, which is the blood vessel. But because we have not stained it, we are putting it in black and white, okay? But actually, this is blood vessel, right? They are saying Haversian canal, they are saying, right? It says blood vessel, right? So what and all will you label here? The lacunae, remember here. So each of this you can see here, lacunae, it has something inside. So actually what it is inside this one, they are calling it as osteocyte, okay? And in between all these fibers, they are saying it is canaliculi, okay? So you have to draw the structure. For what and all you will draw the structure? If they ask you transverse section of bone, or if they ask you compact bone, or if they ask you structure of osteon. For all these things, you will draw the same diagram only. So practice it very well, okay? Compact bone, transverse section, osteon. All this you'll have to draw this only. So where is this actually? So this is osteon. So many osteons are there here in the compact bone, okay? So many osteons are there. And the Haversian canals are where? Like this they are. They are perpendicular to this, whatever you have drawn here, okay? So this is the not to repeat again, but anyways, transverse section of bone. So if they ask you histology of compact bone, same thing you will draw. If they ask you for osteon also, same thing you will draw. Okay. But what if they ask you the longitudinal section of bone? I will take a bone now and longitudinal section. So in that, what will you see? You will see the Haversian canals, correct? And perpendicularly, you will see the Volkmann's canals, correct? So you will see what and all? You will see the Haversian canals, which you're seeing here, the Haversian canals, right? This is again black and white image, remember, because we have not stained it. Haversian canal. This is Haversian canal and this is Volkmann's canal, okay? Again, same thing, lacunae and canaliculi, okay? Canaliculi and lacunae. Lacunae, lacunae, canaliculi, 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 okay? So this is the image when you see under the microscope, bone LS. Longitudinal section. So now let us look at this second question here. People, we have moved on to what? The second question, second topic here. The sensory and sympathetic ganglia. Okay, so they are asking you, compare it and contrast the microscopic structure of sensory and sympathetic ganglia. So basically you should know that they're asking you even to write things. So it is 5 marks. They're asking you to compare these two also. So you'll have to compare. Okay, so what is sensory and sympathetic ganglia? So what is this you're looking at here? Spinal cord, ventral root of spinal nerve, and where do you have the sensory and where do you have the sympathetic? Basically, the dorsal is sensory. Okay, so that much you can remember. So when we compare, so first let us look at this. This is the spinal ganglion or the sensory ganglion or the dorsal root ganglion. So here you can see the word written here. This is dorsal root ganglia, spinal. Yeah, this is sensory. Okay. Then you have what? The sympathetic. Now they are asking you to compare and contrast, right? So that is what you have to know here. So let us look at this one. So you can look at the actual nerve here, kind of a thing. They have taken a cross section here. Anyways, what you have to focus on is that the nucleus is centrally placed, right? And it is circular. And around it, what do you have? You have the cytoplasm, right? Same thing they have labeled in this diagram. You can see here, centrally placed nucleus and axon. Then you have the cytoplasm around it, which they have labeled here. This is a cytoplasm. And then around that outside, they have shown the satellite cells, okay? Satellite cells, okay? So this is spinal ganglion, sensory ganglion, dorsal root ganglion. Now, histology-wise, when you want sympathetic ganglion, look at sympathetic ganglion. The cells are not grouped together; they are here, there, etc. Right? And you can see that they have taken a cross section here. So what you're seeing is it is star-shaped. They are saying this is not complete circular like the other ones. So these cells are not completely circular. These are star-shaped. They are saying. Okay, and there is an eccentric nucleus. So this is the cytoplasm. There's an eccentric nucleus, and you have branched fibers. They are saying here branched fibers, and then you they have labeled this as a sympathetic ganglion. Okay, so this much is enough for you. Histology wise, if you want to know, this much is overall. Okay, let's move on. So we are done with 2 questions. So now shall we move on to the third one? Great. We are just looking at it very high level. Can they ask you sarcomere? Sarcomere is kind of a microscopic structure, though it is not histology. We have not studied it in histology as such. Just know about sarcomere, okay? How skeletal muscle will contract and relax, etc. Okay? Actin, myosin. So we will move on now to the 4th question in histology, or 3rd question you can say. Okay, microscopic structure of cardiac muscle. So they are asking you what? Microscopic structure of cardiac muscle, the heart. Heart muscle they are asking you. So what do you know about heart muscle? Say heart muscle, it will be branched. Very good. So very, very important for you. Under microscope, you will be able to recognize the heart muscle, right? So how will it look? So this is your heart. Here is your muscle of the heart. You can see that there are intercalated discs. If you're not noticing. So here you have intercalated discs, right? And then you have branching. Very important to notice under the microscope is the branching. You can see here this fellow is coming here and then he's going like this and then he's going like this. So there is branching. So most important thing, intercalated disc and branching. So answer this question. Which of the following is a microscopic feature of cardiac muscle? Eccentric nucleus. I don't see an eccentric nucleus. Fusiform shape. Is it fusiform shape? Fusiform means what? Something like this, is it? What is fusiform shaped? Intercalated disc. Yes, that is correct. And it will have branching. So this no branching is wrong. So answer is intercalated disc. Okay. Very good. Histology can come in form of MCQ also. So look at the answer, the comparison here of all the 3 muscle types. Okay. So you have the striated, non-striated and cardiac. Cardiac means what? Heart muscle. So basically it is branched. Most important word, intercalated disc. Okay. Nothing else that you have to remember much. It is uninucleated. You saw that it has 1 nucleus, right? Then you have faint, light and dark bands. Then that's enough. Okay. Nerve supplies, autonomic nervous system. Obviously you cannot control it, right? This, you become anxious, it will start beating fast. But otherwise, it is autonomic. That's all you have to know. Okay. Let's move on. Okay, we are going to pass histology questions. Yes. The next one here is serous and mucous acini. What is it? Serous and mucous acini. Compare and contrast. Okay. So basically comparison and contrast. That means you have to say similar points and dissimilar points both. Okay. You have to draw a table and put each on others on this side and this side, and then you have to compare and contrast. Apart from that, you have to draw a diagram also. Okay. So diagram only we will learn for now. That will give, then we will make that into table also. Serous and mucous acini. Okay. Let's go here. So they've asked again salivary gland, serous gland structure. So they have asked this again and again. So remember. So this is the serous gland. This is how it looks under the microscope. So let us look at the differences. Here you have serous acini, mucous acini, and the one that combines both. So what are you seeing here? In the serous one and the mucous one makes mucus, I'm thinking, right? Mucus, it makes mucus. Okay, so it has the mucous one. We are seeing that it has some flat nuclei, flat nuclei. Okay, The serous one has round nuclei. Okay. Then pinpoint lumen. So the lumen is just pinpoint. It is not a very big lumen, a small lumen. But here you see the lumen is big. I'm seeing a big lumen. Okay, great. Now, next, spherical nucleus. We already said that, right? Spherical nucleus and flat nucleus. Okay, this one. Basal basophilia. Wow, what a word. Basal basophilia. So it's a little blue here. Is it basal basophilia? Even this has basal basophilia. Oh. Yeah, this has basal. This has pyramidal cell. This is also pyramidal cell only. I'm seeing pyramidal cell in both. In both, I'm seeing pyramidal cell. But basically, remember nucleus structure. Nucleus is round, nucleus is flat, pinpoint lumen, and large lumen here. Okay, done. And there's a lot of mucus secretion they've shown here that we can do nicely, right? We'll also draw here. Lot of mucus secretion in the mucus acini. Okay, moving on. See this mucus acini, right? Full of mucus only. Okay. And then here, this diagram is so dark. Can you increase some brightness, please? Oh, thank you. So here you can see mucus salivary gland. This is drawn by me in the book. Hope it's drawn correctly. So you have the mucus acini. In the middle, there is that part you leave that is not there. There's only one circle is mucus acini. In the middle, you have the lumen where it is pouring all the mucus. You have flat nuclei, right? Basal flat nucleus. Then what else was there? Basal basophilia, they said. Then one more thing. It's not a pinpoint lumen in the middle. Okay, it's a huge lumen. Okay, great. In spotter, if they keep off something like this, then you have to recognize that it is mucus and serous and hence it is mixed maybe. Okay, fine. Next question. Do you want a break, people? Is it too much? Okay, relax. Excellent. So next question, microscopic structure of lymph node. See, this is a microscopic structure. Okay, so we'll take it as histology. Okay, so this is the diagram taken from very beautiful book, I'm sure. So very easy to remember to draw this. Okay, I will draw this and show you. It is so cute easy to remember. Draw one tree. Okay, in school you will draw one tree. Like this one tree you will draw. Okay, and here this one input pipe. Okay, and then here this one output pipe. Simple. Okay. And always everything will have blood vessel, no? So you put one red here and one blue here. This tree is getting a lot of nutrition from the ground, right? Red and blue. But something from top is coming, some rainwater. And then it's filtering and it is going down. Okay, so this is our lymph node. Inside lymph node also some important things are there. Okay. Germinal center germinal center. Yeah, here we found it. See? Germinal centers full of cells, right? Germinal centers everywhere. You draw one like this, and outside one capsule you say. Okay, this is capsule you say. Okay, then what do you have in the middle? In the middle there is some medullary sinus. Medullary sinus, guys, are you bored? Medullary sinus is there here. What is there? This one input. Okay, and what they are saying is efferent lymphatic vessel. Okay, great. Then there's something called as primary follicle and secondary follicle. Okay, what is primary and what is secondary? Okay, so now look at this diagram hand drawn means how it will look. I don't like this. I would like to draw the tree only. But anyways, just notice here you have germinal centers, you have capsule labeled here, you have lymph nodule. This is blood vessel, medullary sinus, and that's enough. Okay. Thank you. No, still video is going on. Hold on. So here you have some structure of lymph node which people drew. Promise people only few more questions. Okay, we'll finish fast also. Histology will get completed. Okay, what you say? Smile. And always, when they ask you histology questions, you also write some clinical anatomy. Okay. In the end, you say, what is the importance? Now, you studied about lymph node, like lymphadenopathy in tuberculosis or anything like matted lymph nodes in tuberculosis. All that you can write, okay? Types of, microscopy of hyaline cartilage. Microscopy of hyaline cartilage, okay? So, what are they asking you here? Cartilage. So, this is the diagram, how it looks under the microscope and how will you draw it. So let's just look at how the cells are drawn here. Like this and like this, inside this. and in that there's a nucleus and nucleus. So like this they have drawn, in this they have drawn 4 cells. Otherwise there are 2, 2, 2, 2, 2, and only one is there in this. So what is there inside? This is a chondrocyte. So here also you have one chondrocyte, one chondrocyte. To draw, you will draw one line here, and then you will draw a lot of cells here. Then you will draw a lot of matrix here. Within the matrix, you will draw those cells that we told you. Okay. So here you have the perichondrium. Remember, it is not cardium. Chondrium. Chondro is what? Cartilage. Perichondrium. Perichondrium. Then you have chondroblasts. Yeah, they are the new cells. Chondroblasts. They will become the chondrocytes, right? Blast will become cyte. So you have the chondroblast, then you have the chondrocyte. Sometimes these sites, no, it's chondrocytes, they are together like this, right? So many sites, and around it there is something. So this is called as a cell nest, right? They are all together. So that is cell nest. Around these cells, there is something, a little matrix you see around this nest, that is called as territorial matrix. Territorial matrix means what? Around it, the territory they are marking. Okay, territorial matrix. But in between, whatever you see here, all this is interstitial matrix. Okay, around these chondrocytes, territorial matrix. This is what? Hyaline cartilage. Can you see all that in this? Can you see all that in this diagram, which is real diagram under the microscope? Shall I zoom it for you? Zoom, 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 zoom. Can you see territorial matrix? Can you see cell nests? Can you see chondrocytes? Then if I go up, can you see chondroblasts? And if I go up, you should be able to see the perichondrium. Okay. What do you think the next question is? Thymus. Yeah, where is thymus? So many times we don't even know where the thymus is. We know where the thyroid is, but where's the thymus? Thymus is here, people. Look at this. Above your heart, directly above the heart, they're showing thymus. Thyroid gland is still here. The thymus they're showing here. Not much I remember about learning about thymus. But anyways, neat label diagram of microscopic structure of thymus. So thymus means how it will it be? Again, purple only I'm feeling because it is one of those lymphoid. Yes, thymus. You look at this. So purpley. Yeah. Now how will you draw the diagram? Zoom, 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 and increase the brightness. Because without brightness, can you see? Okay, that's bright enough. Thank you. So what are you seeing here? Thymus. So thymus, there's a capsule. Very good. And then there's nucleus here they have shown. There is some septae like this. Capsule, septae. Okay. Then you have trabeculae. They're showing here. Then in the middle they have shown some medulla. The center thing is medulla. They are saying. Medulla. So on this side, what you'll have? Cortex and cortex. What you'll have in the middle? Medulla. And on the either side, they've shown the capsule. Okay, this is capsule. On both sides, they've shown capsule. Middle, medulla. And then here they have shown what? Cortex. I told you. Medulla, I told you. Very good. Same thing I'm telling you. Within the medulla, what is there? Very important. Hassall's corpuscles. They are connected. Hassall's corpuscles. Hassall. H-A-S-S-A-L-L. Hassall. Everything is double in this. Okay, it's a hassle to remember this thymus. Hassall. And they've shown some blood vessels here in the septum kind of a thing. Okay. So this is the microscopic structure that you will have to draw. Neat labeled thymus diagram. Microscopy. Okay. Now the next question is about this diagram that you're seeing here. What do you think it could be? Brain. Oh, little below that. What is that? Yeah, pituitary gland. Very good. Microscopic anatomy of pituitary gland. They've asked for 5 marks, so please draw a diagram and explain also. Okay, so where is the pituitary gland? Here. Do you want me to zoom and show where pituitary gland will be? Here. Okay, pituitary gland is here. Okay, hypothalamus, then pituitary. Hypothalamus is the master of master. Pituitary is the master. Okay, if you draw this diagram also to explain anatomical position, then they'll be very, very happy and give you good marks. But you should draw one diagram roughly. So you will also have a that kind of a layout in your brain. Okay, so here you can see pituitary gland microscopy. This is so different. You can see here one type, here one type, and here one type. Why is it like that? Because here you have one type and here you have one type and there's some gap here and one type. Exactly like this diagram we have drawn in this book. So we need to understand it. So let us zoom. But before we zoom, let us see. This is posterior pituitary. This is anterior pituitary. In between you have the pars intermedia. And there are some other terminologies here. Look at this. Pars tuberalis, and pars distalis. Can you say pars tuberalis?
Pars tuberalis.
Tuberalis.
Pars tuberalis.
And pars distalis.
Pars distalis.
Thank you. Okay, so now let us look at this. This is nothing but the pituitary gland or the hypophysis cerebri. Okay. So basically, what are you going to draw here? Let's go part by part here. They have shown nucleus of pituitary. I think they wanted to keep some cute name for this pituitary gland sites, so they have called it as pituit sites. Okay. Then you have some unmyelinated axons. Then they are saying capillaries. They have shown some capillaries here with some ... And then, okay, so where were we in pars intermedia? Here they are showing you the cysts. What are they showing you? Cysts, right? Which have colloid. What do these cysts have? Colloid, just like some thyroid colloid. They are talking about cyst containing colloid. Okay. So this is lined by cuboidal cells. Obviously, there is colloid in the middle. These are cysts. There are cuboidal cells and colloid in the middle. Okay. So colloid follicles. Okay. Then they are saying that there are some basophilic cells in clumps. Basophilic means blue. So basophilic cells in clumps. Okay. Now coming to anterior pituitary. So pars distalis, anterior. So what and all is there here? So many cells you can see here. There's a lot of cells here, right? So you have 3 types of cells, if I'm not wrong. 3 or how many types of cells? Chromophobes, acidophils, right? Chromophobes hate color, so they are white. Chromophobes. Acidophils, they like acidity, looks like red they are. Then you have then you have basophils. Sorry, wait. What and all do you have? Chromophobes, acidophils, and basophils. You have basophils, they've shown in blue, right? Basophils. Acidophils you show in little pink. Chromophobes, no color. Okay, lot of cells here. And then here you have middle, some gap. So sinusoids. So in the middle you have gap, sinusoids. And then you have some blood vessels, capillaries. Okay. So what and all you have? 3 types of cells in the anterior pituitary, chromophobes, acidophils, and basophils. Chromophobes, acidophils, basophils. Okay. Chromophobes are more. So remember 50% of them should be colorless. Here in the cells, and acidophils are 40%. You'll have to make 40% pink and 10% will be basophil. Okay. What will you write for clinical anatomy? The anterior pituitary, you will make so many types of hormones and all that it will release. And posterior will give you only 2 things. Which are those 2 things? So here you have ADH and oxytocin. Okay, oxytocin is coming from posterior pituitary, remember, and the other one is ADH. Only these 2 are posterior. Everything else is anterior. So clinical anatomy, clinical knowledge, little you have to write. That's why you're reading the histology, right? Okay. And pituitary, it is, the spelling is very weird. Okay, it's pituitary. Pituitary. Like that, if you remember, it's better. Okay, pituitary. Okay. I think this is the last question, guys. Cornea. This is cornea. So this is outside and this is inside. So basically, from outside, if you go, what and all layers will you get? Cornea. First of all, where is cornea? Cornea is your outermost part of your eye, if you remember. So this is the eye, right? So you're looking at it from the right side view. Okay, so here you have the cornea, right? Cornea. So they have taken this, cut this part, okay, and they've put it under the microscope. You can see here. Obviously, it will be, won't have a cornea now. Okay, fine. So this is light. Okay, that's why we have shown here. This is light in yellow. We have shown this. Okay, so here you have the microvilli on these cells. So here you can see here they have shown some projections here on these cells. There are some microvilli. Okay, so this is how it starts. This is where the light first falls. Okay, there are some microvilli. There are some cells here, basically, remember, and on that there is some microvilli. Okay, and then you have some cells here again, and then you have some cells here again, and in the middle, basically, you have something called as a substantia propria. Okay, oh, you can't see what I'm writing. Okay, wait, I'll choose a better color. Substantia propria is there. Okay, so basically, what are the main layers you should know? Cornea has many layers. Okay, so look at this here. You have here, you have the epithelium. What do you have here? Epithelium. Then you have the Bowman's membrane. Okay, here you have what? The Bowman's membrane. Then you have substantia propria, and then you have some Descemet's membrane. Why you should know this, because, okay, so there are so many other layers, okay. Pre-Descemet's membrane is there, called as Duas layer, which is a new discovery, it looks like. So pre-Descemet's membrane, you have the Duas layer. So you have to know all these layers, guys. You have the epithelium, then Bowman's membrane, okay. Bowman's membrane itself acts like the basement membrane. Then you have the substantia propria, then you have the Duas layer, and then you have the Descemet's membrane, okay. Behind that, what do you have? Descemet's membrane, they're calling endothelium, okay. Fine. Why do you think this is important? Why cornea they're asking? That's what I'm asking you from that time, right? See, basically now because of LASIK and all that, right, they will do laser surgery and they will make sure that you can see properly, right? So they will adjust the curvature of your cornea. So how many layers get affected? So they will basically lift off this epithelium, right, if I'm not wrong, and basically what they should be reducing is the stroma, that is the substantia propria, I'm thinking. So you can understand, right, how important cornea structure is, and what is there behind cornea? Aqueous humor, okay. So can you see the layers of cornea? Yes, ma'am. Epithelium, then we have the Bowman's membrane, right? Then you have the substantia propria or the stroma, then you have Duas layer, then you have the Descemet's membrane, and then you have the endothelium. Thank you so much. Learn how to draw this, okay. You have some cells here, then you have some hexagonal cells here, then you have some columnar cells here, then you have some Bowman's membrane, then you have substantia propria with some cells here, then you have the Duas layer, then you have the Descemet's membrane, and then the endothelium, okay. So whole of histology, important questions we have covered. So just before your histology practical or just before going to your anatomy final theory paper, please revise, okay. Thank you. Bye bye.